Hello and welcome to Chapter 3, Cells and Their Functions. When we look at Chapter 3, there is three different parts I want you to look at. The main one is cell structure and the organelles, or the parts within the cell. That's important. We're going to look at transport in terms of passive and active transport. And then you're going to look at some cell processes, protein synthesis, such as the transcription and translation, and also cell division of meiosis and mitosis. Okay, so first thing we go through is a little bit on microscopes. There are three types of microscopes we're concerned of: the light microscope, the transmitting electron microscope, and the scanning. When you look at a microscope, you can see the difference based on the picture. First off, light microscope is not great at magnification. You can only get to a magnification about 200, and it's pretty flat. When you get to, so here's the cilia here, this one's in larger magnification, so it's the electron microscope. You can tell the difference between our transmitting and our scanning. Scanning is more three-dimensional. Okay, light microscopes use light for resolution, and scanning electrons use electrons for resolution. There's a little bit more about microscopes and cell anatomy on a video called Discovering the Cell, an older video, but it's very good. I'll put that link up later on. Now, cell structure is important. When you look at cell structure, the cell is very organized. There are organelles, or bits, inside of the cell. It's important you're aware of all these structures and their main function. So when you look at the structures, they're in you've got the nucleus, which is the main control center of the cell. This has the genetic information, and it regulates what proteins are going to make. What proteins are going to be made. The plasma membrane regulates what goes in and out. The endoplasmic reticulum is a site for packaging and transport. There's two types of endoplasmic reticulum. There's rough, which has ribosomes on it. Ribosomes make it rough. And there's smooth endoplasmic reticulum. The smooth endoplasmic reticulum is more involved with lipids, packaging and protein lipids. And the rough endoplasmic reticulum is proteins. Then those will both be shipped to the Golgi apparatus, and the Golgi apparatus is a site for further modification and transport. When you look at energy in the cell is run on ATP, and the ATP molecule comes from the mitochondria. This is the site where it's made. So if you think of the mighty mitochondria, and a mitochondria has finger-like projections, unlike the plant cell, which has chloroplasts, and the chloroplasts kind of look like stacks of pancakes. So finger-like projections is chloroplasts. Sorry, mitochondria. Then you have vesicles, and vesicle is a transport sac. If they're larger, they're called vacuoles. If they're smaller, they're called vesicles. Once they get specialized, the vesicle could be called a perisome or a lysosome. All of those are used for transport. Then you have your centrioles. Centrioles are used in cell division, which we'll look at when we get to mitosis. And that's a pretty rough estimate. There are tables in the textbook which describe each one. When you look a little more at the plasma membrane, the plasma membrane is a phospholipid bilayer, which means it has two layers of phospholipids. Now, phospholipids is this unique structure molecule. If you remember, it's a lipid where one of the fatty acids has been replaced by a phosphate group. That makes the molecule have this non-polar end on the inside and a polar head on the outside, making the molecule ideal for membranes. So you have the non-polar inside, polar outside reacts with water. So it's a bilayer in the fact that there's two layers of it. Inside the phospholipid bilayer are proteins. Proteins can act as channels or they can act as transporters. Okay, there's a bit there on the cell plasma membrane. And as you go through, there is tables in the textbook that describe each. You do need to know the description, and you do need to know the function. You will not be asked to label a cell. However, you do need to know the general description and the function for multiple choice type questions. Okay, please realize when it talks about the inside of the cell, it can be called the cytocell or the cytoplasm. The cytoplasm is everything inside the cell. The cytosol is the liquid portion. Quite often these are interchangeable. Okay. There is great cell diversity, and the cell diversity has to relate to the function. If you want something, nerve cells are long, you've got epithelial cells which are strong like bricks, and you've got muscle fibers which are long, red blood cells which look like little donuts, and this is a sperm cell which is pretty much DNA with a long tail to keep it going. Okay, there are some questions there. Now, when it comes to transport, the purpose of the cell membrane is to transport. And transport is based on something called selective permeability. It's selective in the fact that if you are small and small and not charged, you can go through no problem. But if you you can't travel across if you are large, if you are non-soluble, or if you were charged. So this has to be moved differently. Now, you can be moved in two ways. You can be moved passively, which does not require cell energy, or you can use the energy and move actively. When you look at passive transport, there are a couple of categories of non-active, non-energy requiring, or passive transport. One is diffusion, which is basically simple movement down a concentration gradient. You could have facilitated diffusion, which is down a concentration gradient with the help of a channel. When you look back to the plasma membrane, here you have a channel. This molecule, this green molecule, will move through based on high concentration to low concentration. It needs a channel for it to go through, but no energy is required to get it through. It just moves from high concentration to low concentration. Then, when you look at the movement of water is called osmosis. And the movement, again, is going down the concentration gradient from a place of high concentration of water to a place of low concentration of water. However, when we talk about the movement of water, we more look at not the concentration of water, but the concentration of solute. So when you're looking at it, water moves from a place of high concentration of water or a low concentration of solute into a place of high concentration of solute. So it's like the water wants to move to dilute out the solution. What happens in most situations is the salt is pumped and then water moves to balance out the salt. We'll go into a little more detail on this. I have some videos up on hypertonic and hypotonic and blood cells, and we'll look at that. But when you think of osmosis, think about it in terms of the concentration gradient. Okay, when we look at osmosis on cells, by changing the fluid around the cells, you can change what happens to water movement in the cell. Isotonic is what you want as a normal situation. Iso means the same, so you want the same concentration. Now, notice that water will move in and out of the cell, but it is at the same rate. So the net balance is 0. In isotonic solutions, there is movement of water, but the in balances the out. When you get to hypo, in hypo, the concentration is low and the cell will grow. Low, so grow hypo. This will happen if you have a dilute concentration or pure water. This is a big deal if you are a runner, a marathon runner, and you end up drinking a lot of pure water and not Gatorade. What it'll do is dilute your blood. When it dilutes your blood, the water will want to go from a place of high water concentration or low solute concentration and move in the cell. As it moves in the cell, it will cause the red blood cell to swell, and since they are fragile cells, you will end up with bursting cells or hemolysis. Not good for the blood cell. Now, if you were to put in too concentrated of an IV solution, you can draw water out of the cells. In this situation, an example is hypotonic. In hypertonic, hyper means more, so you have more non-water particles outside in the solution. Water will move from a place where there's high water, low solute, to low water, high solute, and the water will move out and the cell will shrink, and you end up with what's called crenation. Cell movement can also be a result of filtration. Filtration occurs in the kidney, and this usually has a force due to blood pressure. It's blood pressure that pretty much pushes things through, but it is movement from high concentration to low concentration. Now, another way, so that's all passive transport. Passive transport does not use energy in the system, or sorry, extra energy. It uses energy in the system. So there is also active transport. Active transport requires energy. You can have a process where things are pumped, which we'll look at when we look at the ATP pumping in the neuron and the muscle. You can also have endocytosis and exocytosis. Endocytosis and exocytosis use the membrane. Endo means in, exo means out. So when you look at endocytosis, the membrane will actually surround a particle, producing its own little vacuole, and it will pull it in. Endo, in. Exo is the opposite, and this is actually a form of secretion. The vesicle will come, it will fuse with the membrane, and then the material that was in the vesicle will be added out. Okay, so that is movement. I'm going to end the video here. So on part 1, we need to know the organelles, and you need to know about active and passive transport.